Hello, dear students. I welcome you all on behalf of Sri Jayendra Rank Zone classes. Dear student, we are in a continuation of the chapter that is molecular basis of inheritance. And today we are going to deal with the topic that is SGP, the Human Genome Project. Dear students, it is a great project which was done by different scientists or a group of scientists in a very well manner. And by this project, several disciplines in a science arised in the field of genetics. So, dear students, if we talk about the genetic makeup of an organism or an individual, we know that this genetic makeup lies in the DNA sequence. And the differences in the two individual will naturally be reflected in the differences of their nucleotide sequence. Means if we compare the two individuals, then we will see that the differences in them are reflected in the differences of the nucleotide sequence in the DNA. They can be known only if the entire human genome is mapped. Means we can identify these differences if we know the entire nucleotide sequence of the particular human genetic constitutor or human genome. And with the establishment of the genetic engineering technique, it was possible to isolate and clone any piece of DNA, and availability of simple and fast technique for determining DNA sequences, a very ambitious project of sequencing human genome was launched in the 1900. Okay, so be remember the first point to sequence the whole genome. See, first you should understand the word genome. So genome, it consists of all the genes present in one set of chromosome. So whatever the genes which are present in one set of chromosome, they combinedly known as genome. So this project is targeted with the sequencing of this genome. So a very ambitious project, very ambitious project of sequencing human genome launched in year 1990. Now, this project, when was it was launched in 1990, it is considered that SGP or Human Genome Project was the international collaborative research program. It is a collaborative research program. Means so many lab of different countries have participated. So many scientists of different countries have participated in this research program. And the goal of SGP was to complete mapping and understanding of all the genes of human being. So it is an international project. So many scientists have been collaborated, and they are targeting to map and understand all the genes of human genome. Now here mapping means what? We have to identify the location of gene, and we have to understand that in gene what is the sequence of this gene, or what is the nucleotide sequence of this. So all the genes together of a haploid set of chromosome are known as what? We can say a genome, and that we have discussed. So when the international collaboration happened, then it was considered as this SGP as mega project. It was considered as mega project, P as mega project, and it was 13-year project. And this 13-year project, it was coordinated by US Department of Energy and National Institute of Health. So initially it was started, means we can say funding was provided by this US Department of Energy and National Institute of Health, and they coordinated this project. Okay. But later on, it is known that Wellcome Trust soon, or we can say Wellcome Trust from UK joined the project as a major partner. So now three are there to coordinate this project. First is US Department of Energy, another one is National Institute of Health, and the another one is the Wellcome Trust. These three are there to coordinate this thing. Now additionally, it was known that additional contributions came from Japan, France, Germany, China, and other countries. Okay. So now this many people contributed, and they are targeting to sequence the whole genome. Means we have to know that at which position which nucleotide is present. It is either adenine, it is guanine, it is cytosine, or it is thymine. Whatever present at the first nucleotide, at the second nucleotide, at the third nucleotide, at the 10th nucleotide, at the 1000th nucleotide, what the this nucleotide is present, that was particularly considered. Now it is known that this project was completed in this project was completed in 2003, and now it is considered that SGP or Human Genome Project has been called a mega project, has been called mega project. So now the question is why it is known as mega project. So it has been called as mega project due to, so the reasons for this is that number one is huge cost, huge estimated cost of this project, and it is to be 9 billion US dollar. So the cost of sequencing one base pair like AT or GC, one base pair to sequence is $3. Okay, so this is the particular huge cost. So the participation for different countries were received and it is considered as mega project. The second reason for the consideration of it as mega project is that very large number of base pair, very large number of base pair, that is 3 into 10 raised to 9 base pair to be identified and sequenced. The third reason for this, that it require large number of scientists, large number of scientists, technicians, and supporting staff. So this is also the one of the reason that the particularly it is a mega project. So first is what, a huge cost. Second is what, large number of base pair, 3 to 10 is to 9 base in haploid cell in one set of chromosome, and large number of scientists, technicians, and supporting staff. So involved. So it is considered as mega project. The other reason is storage of a data generated. Now, why the storage of data generated is one of the particular, because of this it is considered as a mega project, because the storage of data generated, it is known that it required, if we are going to write, it required some 3300 books, and each book with 1000 pages, each book with 1000 pages, and each page having 1000 type letters. Okay, so this is the particularly 3300 books, and each book with the 1000 pages, and each page having the 1000 type letters. If this much we write, then we can particularly store this data. So if we keep on writing, then storage will be also easy, we can do. But after storage is there, analysis is the important. Analysis cannot happen. So however, high speed computer devices or computational devices for storage, retrieval, and analysis of data made it easier, to do so. Okay, so high speed computational devices have been developed which can either easily store, retrieve, and analyze the data, so it will be beneficial for the project to carry out. And the fourth thing which was there, fifth thing which was there to consider it is a mega project, that the retrieval is important, and for this the science of bioinformatics also developed during this period and helped SGP. So these are the what are due to this it is considered as a mega project. So bioinformatics is the branch, if we can say, if we apply computer to this biology or genetics, and we use a computer in this for studying this genetics or in all this, then it is a branch that is known as bioinformatics. That was also developed, and due to all this region, it is considered as a mega project. Now this things after 13 year it was taken to complete this project in completed in 2003. Okay. Now, so before starting this project, they have certain goals. So now we are going to target the goals of SGP. And if we talk about the goals of SGP, what are the goals to this project? So if we talk about goals of SGP, following are the goals of this SGP. And first is identification identification of all the approximately 20,000 to 25,000 genes in human DNA. Second, if we talk, it is to determine sequence of the 3 billion chemical base pair. That is 3 into 10 is to 9 base pair. Okay, we can say. So that 3 billion chemical base pair to make up human DNA means they are present in human DNA. So first identification of all the genes, then determining the sequence of the 3 billion base pair. The third goal was to store the information in database. Storage of information in database. Fourth goal was to improve the tools for data analysis. To improve tools for data analysis. And fifth was the transfer related technologies transport related technologies to other sector such as different industries. Suppose if we get a knowledge of this human sequence, then we can apply it to the pharmaceutical tech companies, and in that industry we can help them to solve the particular some disorder. Some genetic information can help them to solve the disorder, mystery of the disorder, okay, or to treat them. So for this, also transport technology has to be understood. Then the another goal was to solve ethical, legal, social issues, that is ELSI. So to solve any ethical, legal, and social issues means we need to address. Suppose if we are going to dealing with the human genetics, then at that time so many priests and so many people who are religious people who thought that it is illegal. So to solve such ethical, legal, and social issues, this was one of the important goal during this particular human genome project. The next goal is to improve the close association of the SGP with the rapid development of new era in biology, that is bioinformatics. So to improve close association of SGP with the rapid development of new area in biology, that is bioinformatics. Then the next goal was not to solve only the genome of human, but to also particular sequence the genome of other non-human organism. So the another goal was to sequencing the genome of model organism. Okay. Now this model organism, if we talk, they are the non-human organism, and this non-human organism DNA sequence can lead to an understanding, can lead to an understanding of their natural capabilities, of their natural capabilities that can be applied, that can be applied towards solving challenges in healthcare, agriculture, energy production, environment remediation. Okay, so in all this, if we understand the sequence of the non-human organism, that will help us to understand the natural capabilities, and that can be applied to solve the challenges which are in the healthcare. Like we are not able to solve the, we are not able to identify the particularly any antibiotic or any, we can say, solution to any disease, that treatment to any disease, that it can help in this one. In healthcare, in agriculture also, we can improve some of the produce, improved qualities, improved varieties. In energy production also it can help. Now the many non-human organisms such as which are involved, and if we talk about the non-human organism, then non-human model organism are, if we talk about this organism, then they are bacteria. Like if we talk about, write a name of the organisms, the first one we can write bacteria, that is E. coli, that is Escherichia coli. Then second organism is Saccharomyces cerevisiae. This Saccharomyces cerevisiae, if we talk, that is yeast. Then third, we can talk about the Caenorhabditis elegans. If we talk about this Caenorhabditis elegans, then it is a free living non-pathogenic nematode. Okay, and fourth, if we talk of example, then it is Drosophila melanogaster. We know that Drosophila melanogaster, it is a fruit fly. Other organisms are Arabidopsis and Rhizosativa. In this, if we talk, they have different base pair and number of genes in them. If we talk about E. coli, the E. coli have 4.7 million base pair and 4,000 genes. Same way, if we talk about yeast Saccharomyces cerevisiae, they have 12 million base pair and 6,000 genes. Then Caenorhabditis elegans, if we talk, they have 97 million base pair and 18,000 genes. Whereas Drosophila, if we talk, they have 180 million base pair and 30,000 gene, 13,000 gene. And after this, another organism, if we talk, that another organism, that is the fifth one, if we talk, that is Arabidopsis, that is plant. And in this, 130 million base pair are there and 25,000 genes are there. Whereas next example is Oryza sativa. Oryza sativa is nothing but a rice. And in rice, it is known that there are 430 million base pair. And in 430 million base pair, 32,000 to 50,000 genes are there. Okay, so these are the non-human model organism whose sequences, their target was to sequence the genes of this also. Okay, so these are the all the goals which was decided by the scientist of the Human Genome Project initially before the starting the project. So after understanding these goals, now we are going to talk about the methodology which was used by scientists to this human genome sequence. So next is methodologies. And in these methodologies, two major approach was there. And these two major approach include the first approach, that is EST, or we can say the ESTs. ESTs is expressed sequence tags. So this approach include to identify or identifying all genes that are expressed as RNA, means to identify the sequence of all the genes which are expressed as RNA, means which are undergoing a transcription, means which include the coding sequences which has a coding information. So that is the first group of scientists which was thought to continue the sequencing like this. Not all DNA sequence get all DNA gets sequenced. Only the coding sequences should be sequenced. And the another group of scientists was talking about sequence annotation. And now, what is sequence annotation? The sequence annotation means sequencing of the whole set of sequences of the whole set of genome that contain all the coding, all the coding and non-coding sequences, and later assigning different regions in the sequence with their function. So identify all the coding and non-coding sequences, sequence whole the genome, DNA of whole genome, and later they assigning the different regions whose function can be identified with their function. So this two idea was there, but later on majority scientists preferred the sequence annotation. So now how the sequencing was taken place. So for sequencing, it is known that what done means for sequencing, what is the methodology. So for that, first the total DNA from cell is isolated. Means the DNA extraction is done first. All the DNA from the cell is isolated. Then after the isolation of DNA, this DNA converted into random fragments of relatively smaller size. Okay. We know that DNA is a very long polymer, so there are technical limitation in sequencing a very long piece of DNA. So the small fragments or a smaller size fragments have been created, and these fragments cloned in suitable host using specialized vectors. Here vectors which are used, that are the plasmids, vectors are the carriers, and the plasmid, they will take this particular DNA fragment. So this cloning done in this particular vectors, and this cloning result into amplification. Amplification means increase in number of copies of each piece of DNA. So if piece of DNA was copied several times and their number of copies increased by this amplification. So why this amplification is necessary? So it is known that amplification is necessary because these fragments could be sequenced with ease. So for this amplification is necessary. Now it is known that the commonly used host were bacteria and yeast. And the vectors which was used here were called BAC, that is bacterial artificial chromosome. So if we take, suppose this is the bacteria, this is the DNA of bacteria. Now if we take a BAC, that is a plasmid artificially designed, and in that we add the DNA fragment which is going to be sequenced and inserted or cloned into bacteria. Now in bacteria, it will use its machinery and several copies of this vector will be produced. Same way, another vector is YAC, that is yeast artificial chromosome and such like. The in yeast artificial chromosome fragment is inserted and that yeast artificial chromosome plasmid is inserted into the host, that is yeast, and where the replication and copying are copies are produced. So now when these copies are produced, we have so many fragments, so we can perform this sequencing easily. So now it is known that the fragments were sequenced using automated DNA sequencers. Automated DNA sequencers are the particular sequencing machines, and it is known that this automated DNA sequencer on principle of method worked on principle of a method developed by Frederick Singer. Okay, and this Frederick Singer is developed a method for the sequencing of DNA. Apart from this, he Singer is also credited for developing method for determination of amino acid for determination of amino acid sequence in proteins. So Frederick Singer was worked two major things. One is the DNA sequencing, and in the another is the amino acid sequence in the protein. And so based on this, it is known that the Sanger was awarded Nobel Prize, two Nobel Prize for these two works. Now, and this through this, the whole sequence of this human genome was achieved, and they have received. When the sequencing they have done, they observed that the sequence of chromosome 1 was completed in May 2006, and this was the last of the 24 chromosome, that is 23 chromosomes, 22 chromosomes and one X and Y chromosome. Out of this was the last to be this chromosome 1. It was last of 24 chromosome to be sequenced because it is a large chromosome. Otherwise, other all chromosomes have been sequenced till the 2003. Okay, in such a way, by using this methodology, the sequencing of the human genome was done. And okay, now, so after the sequencing was completed, they have achieved so much data, and they have analyzed it, and they have shown that some silent observations which are drawn from this human genome project. So now next we are going to talk about the salient features of this human genome which was sequenced. And if we talk about the salient features, these are: number one, he observed that in human genome, the human genome contain 3164.7 million nucleotide bases. So human genome has this many nucleotide bases. Second salient feature was that the average gene consists 3000 bases. But size varies greatly, and it is known that largest known human gene is dystrophin gene. And this dystrophin gene has 2.4 million bases. And the smallest gene is TDF gene, that is testis determining factor gene, and this has only 14 bases. So this was the information which was received after the study of the human genome, and this is the second salient feature. After this, the third salient feature, if we talk, the third salient feature is total number of genes is estimated at 30,000. And this 30,000 genes are present in human genome is what discovered, but it is a much lower than the previous estimate of something 80,000 to 1,40,000 genes. And it is known that almost all, that is 99.9% nucleotide bases are exactly the same in all people. The fourth function or salient feature is that the functions are unknown for over 50% of discovered genes. Fifth is less than 2% of genome codes for proteins. The sixth is repeated sequences make up very large portion of human genome. Then seventh is repetitive sequences are the stretches of DNA sequences that are repeated many times. So repeated sequences are there are stretches, there are segments of DNA sequences they are repeated many times, sometimes 100 to 1000 times. They are thought to have, it is known that Repeated sequences thought to have no direct coding functions, but they shed light on chromosome structure, dynamics, and evolution. Then eighth silent feature is chromosome 1 has most genes, that is 2968, and Y has fewest, that is 231 genes. The ninth salient feature is scientists identified about 1.4 million SNPs, and SNPs are nothing but single nucleotide polymorphism, which are also pronounced as SNPs. 1.4 million locations where these SNPs are observed, and these SNPs are what? That are the single base DNA differences occur in humans. Means if we compare one humans to the another human, there are 1.4 million locations where you will find a difference of a single base pair, okay, or single base. And this information of single nucleotide polymorphism promises to revolutionize the process of finding chromosomal locations for the disease associated sequences. Means we can identify the sequences which are disease related. Means due to the mutation in that sequence, the particular the disease can be recovered. So these are the salient features of the human genome project. Okay, dear students. So now, if we talk about this, that is the salient feature which we have discussed, that all derived from the human genome project. Now we are going to talk about the application. And if we talk about application and future challenges, then the first is the completion of the first phase of human project has to be compared to discovery of antibiotics. So first application to compare the human genome to discovery of antibiotics, because if we compare this, it has opened a vast database of knowledge about the various aspects of human genome, because human genome is a vast data, so it can apply to the different things. Second is soon we shall map all genome. Another application is soon we shall map all human genes and all sequences and all the different types of genes, we can sequence them as well. The third is there are more than 12,000 genes that cause common cardiovascular, imbalance, endocrine diseases like diabetic, Alzheimer disease, cancer, and other neurological ailments. So after taking this snapshot, it will be possible to know the method to alter them and remove the possibility of the disorder. So to help in treatment of disorders by altering the genes which have a defect. Then fourth is single gene defect produce a number of hereditary disease. So the hereditary disease can be corrected. Then fifth is this human genome will help us, or it will be possible to study the interaction between various gene protein as well as the mechanism of formation of tissue, organ and tumors, or switch over to different developmental stages. So it can help to study different developmental stages. And that the sixth is that it holds a promise of healthier and longer living. So it holds promise of healthier and longer living, means we can apply this to the different industries and that can help us for longer living. The designer drugs can be found and genetically modified diets according to the needs of individual human being. Okay, so this is all about the what the application of the future challenges. Okay, so this is all about the human genome project. We have studied why it is called as mega project, what are the goals of this process, what is the methodology, and what are the gains after this human genome, that is the salient features of the human genome. And then we have also discussed the application and the future challenges. So goals, methodology and the salient features are more important with respect to the exam. Okay, so I hope you have understood this. Okay, thank you.